Well, thank you everyone for joining us today for our MPRINT webinar series. Today's talk, we are pleased to have our colleagues from University of California, San Diego Center of Excellence for Therapeutics, Jeremiah Momper and Raymond Suhandynata. I'm sure I said that completely wrong. So sorry, Ray. We appreciate you giving the talk today on behalf of the Pharmacometrics and Analytical Core at San Diego on the role of quantitative bioanalysis in advancing research in maternal and pediatric therapeutics inside the LC-MS black box. With that, I'll turn the meeting to you.
All right, Sarah, thanks so much. Thanks for inviting us to speak today, and we appreciate everyone for joining. As Sarah said, we'll be talking today about quantitative analytical chemistry and its role in maternal and pediatric therapeutic research. As a little bit of background, this presentation initially was structured for the MPRINT trainees. So some of the material may be a little bit basic for experienced investigators, but nonetheless, I hope it will be useful for anyone that's involved in clinical research or pharmacometrics that deals frequently with drug concentrations and biomarker data, but maybe doesn't have the best understanding of how those data are generated from a laboratory perspective and what are some of the key questions that you should be considering when you interface with laboratories and have them work on samples for you. So next slide, please. A little background about who we are and what we do within the pharmacometrics and analytical chemistry core at UCSD. So we are one of the MPRINT Hub scientific cores. And although we're based at UC San Diego and we support the UC San Diego CET, Center of Excellence in Therapeutics, we're also available to support projects across the entire MPRINT. And that includes support for projects that are funded through MPRINT, including the Opportunity and Support Pool projects. So for example, if you're a trainee and you're interested in a prospective study, or perhaps you have stored samples in a freezer somewhere that you could leverage for a new project, we are available to help with those types of assays to drive the research forward. In a nutshell, we're involved in targeted quantitation of drugs, metabolites, and biomarkers, and we have a lot of experience working with small sample volumes and heterogeneous matrices, and we'll talk about that today as we go through. Our analytical footprint has grown quite a lot in recent years. I won't go through everything that we have in the lab, but we're fairly well equipped with LC-MS instrumentation. We also have a variety of GC-MS instrumentation and ICP-MS that's pretty specific for measuring heavy metals, but it's very good at doing that. We also have a MALDI-TOF biotyper and a Q-Exactive Plus, which is a high-res instrument used for identifying unknown compounds. Next slide, please. And our core within MPRINT really grew out of an existing laboratory that we have, the UCSD Pediatric Pharmacology Lab, or PPL. And we're one of the pharmacology support labs within the IMPACT network, an acronym that's likely familiar to many of you. That's the International Maternal Pediatric Adolescent AIDS Clinical Trials Network. It's a mouthful. But this network supports HIV-related research in pregnant women and children, and it's an international network. And we've been involved in assays coming from various protocols within IMPACT now for over 15 years and have performed tens of thousands of drug concentration measurements. And our core also provides a good environment for exposure to not only quantitative bioanalysis, but also pharmacometrics and systems pharmacology. And we have a lot of opportunities to link with other training programs, and there's some synergy across programs that we have, including industry-supported postdoc fellowship that we have that Pfizer funds. We have an NICHD T35 program that supports summer research students for PharmDs and MDs, a critical path grant with FDA's Division of Pediatric and Maternal Health, as well as the Office of Clinical Pharmacology, and then a newly funded Perinatal Health Center of Excellence grant with Dr. Albiwardi at the FDA, and that's looking at pediatric drug safety and the application of quantitative systems pharmacology. So a lot of opportunity to to link with other programs, and certainly encourage all trainees to reach out if there's ways that we can support your research. Next slide, please. Okay, so jumping into the meat of the presentation, bioanalytical methods, of course, quantitatively determine concentrations of drugs, metabolites, therapeutic proteins, and biomarkers in different biological matrices. And the use of validated analytical methods ensures that data are reliable and address certain key questions. For example, does the method measure the intended analyte, or are there other interfering analytes that we need to be aware of? What's the variability associated with those measurements? What's the range in measurements that provide reliable data? For example, the lower limit of quantitation, which is an estimate of how sensitive the assay is, and on the other extreme, the upper limit of quantitation. So what's the range of those, and where can we reliably measure concentrations? And from a practical point of view, questions about how sample collection, handling, and storage could affect data reliability. So basic questions such as how do samples need to be stored, shipped, how long can we store samples and assay them in the future, those types of basic questions. And then through the lens of maternal and pediatric clinical pharmacology research, bioanalytical work here is really associated with some unique matrices that we often don't see routinely in other places, including measuring concentrations in breast milk, cord blood, capillary blood, dried blood spots, and so forth, and particularly related to pediatric research, also dealing with small sample volumes. So we'll talk about all these issues as we go through the seminar today. One of the things that we've been working on that we're very excited about here at UCSD is developing a standardized analytical platform to measure drugs in human milk that accounts for variation in milk composition. And human milk really is quite a heterogeneous matrix. And I would contrast that with blood or plasma, which are certainly heterogeneous from patient to patient, but often not to the degree that milk is. And thinking about things like lipid content, caloric density of milk, those vary significantly, both from mother to mother, but also even during individual feeding sessions, for example, in the morning versus in the evening. So we're working on developing a platform where we measure drug concentrations in milk and not only correlate those with milk itself, but also these individual factors within milk composition that may explain some of the variability to give a better perspective and more robustly predict infant exposures and conversely inform trial design and sample handling requirements for future clinical lactation studies for individual drugs. And these would include things like lipid and protein content, milk phase, for example, fore milk versus hind milk, and then stage of milk production, colostrum versus mature milk. Just one basic example, we recently measured the concentration of THC, tetrahydrocannabinol, in self-reported cannabis users, lactating women who were reported cannabis users. And you can see on the right-hand side, the concentration of THC varied significantly, and it did depend upon the fat content of the milk. So when we defatted the milk, original versus skim, so to speak, you saw a dramatic reduction in the THC concentrations. And that's what you'd expect for lipophilic compounds such as THC, but nonetheless underscores the importance of better understanding the matrix as it relates to contextualizing data when you have milk concentrations. Next slide, please. Also, as we've been working in milk, one of the things that's been important for us is defining for individual studies a useful lower limit of quantitation for clinical lactation studies. And often these types of studies aim to identify a relative infant dose or an RID. And one of the things that's been useful for us is thinking backwards in terms of what's a concentration in milk that's actually impactful and would be a safety concern. In other words, the lower bound of drug concentrations in milk that indicates a negligible infant exposure through breastfeeding, and using that to inform analytical method development, in particular, the LLOQ. So for example, the daily infant dosage for a particular drug in terms of milligram per kilogram or weight-based is often given by the average concentration in milk, which we can determine analytically, multiplied by a typical milk intake, which is generally assumed to be 150 milliliters per kilogram per day. And that gives an RID as stated here in this, this simple formula. And the WHO has put forward RID values that are associated with presumed safety being acceptable, or where caution is warranted, or where the degree of exposure is unacceptable. And that's generally less than 10%, between 10 and 25%, and over 25%. So in a recent project, as an example, we measured remdesivir and its metabolite in milk from lactating women with COVID-19 who are receiving remdesivir for clinical care. And so thinking about what LLOQ do you want to target, we worked backwards and thought about what's the concentration in milk that's actually going to be important. And we decided on 100 nanograms per mil, below which you can estimate that the infant exposure would be less than 1% of that of the mother. And so certainly from an analytical perspective, we could have driven this further. 100 nanograms per mil is a fairly high concentration. We probably could have worked on this assay for another few months and got it down to the low nanogram per mil, but it doesn't change the clinical observation. In other words, if you know that the concentration in milk is less than 100 nanograms per mil, and relative to the mother's dose, that's insignificant, that can drive a little bit in terms of how we structure the assay and what level we shoot for the LLOQ. Next slide, please. Now, other issues that we routinely deal with in pediatric studies in particular would be capillary blood sampling. Many of you are likely familiar with this, and for clinical reasons, this is often done soon after birth, even for infants born with jaundice, where we're looking at bilirubin. So capillary blood collection via heel stick can also be a useful collection to facilitate PK sampling in newborns, but we would caution a few areas of not concern, but things that you need to keep in mind as you analyze data. The first is that heel stick samples can contain some interstitial fluid, and this can have a dilutional effect on drug concentrations. So it's usually good to make sure that nursing staff are performing these appropriately. Usually it should be done on a warmed heel, and blood should be free flowing, as opposed to putting a lot of pressure and squeezing to get blood out, which can cause hemolysis and additional dilution and interstitial fluid in those samples. Also, capillary blood reflects more arterial blood than venous blood. So prior to distribution equilibrium, there can be differences between arterial and venous concentrations. Not such an issue if you're looking at drugs that are dosed chronically upon steady state, but can be something to consider for single dose studies. Next slide, please. So just one example of using capillary heel sticks, and these data include both capillary heel sticks as well as venous blood draws, but just illustrating some of the utility of what we call washout data from transplacentally acquired drugs to really drive and inform future pediatric studies. So in this example, we looked at the drug elvitegravir, which is used for HIV, but these were infants born to mothers receiving elvitegravir for clinical care. And so we can link the cord blood shown on the left-hand side of the graph with washout samples from individual infants. And these are opportunistic samples, so you see some wide windows here, and that's just the nature of how these samples are collected. But this gives us some information about the washout characteristics and the elimination rate of the transplacentally acquired drug in the infant, and we can use that information to design dosing regimens for newborns. So for example, if this drug were to be used in a future pediatric trial, we can use these data to simulate dosing regimens and try to get close to a regimen that's realistic and a regimen that can be carried forward to a future pediatric trial. Next study, please. Sorry, next slide, please. And this is one example of just that. So we looked at very recently newborn dolutegravir dosing requirements and the relation between maternal dosing prior to delivery. And so this illustrates bringing forward cord blood samples and infant washout samples that are obtained either by venous draws or, as I talked about before, through heel stick sampling. And so we developed population PK models separately for the mom and the infant, and those are linked by the dolutegravir concentration at the time of delivery. We also multiplied that by a cord blood to maternal plasma ratio that was based upon the cord blood, of course. And that allows us to predict a simulated neonatal dolutegravir plasma concentration at birth. And I won't go into too much detail, but in effect, these data directly inform the design of a future pediatric trial for dolutegravir, where we're looking at when did the mom take the dose prior to delivery, and also various different infant regimens that could be prospectively studied. And dolutegravir is metabolized by UGT1A1, which of course is not expressed very well at birth. And so it's kind of a moving target as UGT1A1 ramps up. But nonetheless, the maternal dose prior to delivery does directly impact when we give the infant dose and what dose we give. Next slide, please. And then last thing I'll mention before Ray will take over and talk a little about the LCMS considerations would be just a quick plug to consider for your pregnancy studies, measuring unbound concentrations when possible. Now, this is a catchy title from David Berger and colleagues in the Netherlands, but they wrote bound to measure unbound. And so this is an important issue and something that we've seen certainly in recent years provides greater context for interpreting drug concentrations in pregnancy. There is a dilutional effect on albumin during pregnancy. As volume expands, albumin concentrations tend to decrease, and that has an effect on unbound drug concentrations. It can also affect clearance and bioavailability depending upon the extraction ratio of the drug that you're interested in. So a lot of components and a lot of moving parts here, but these data do provide, or unbound data specifically, do provide greater context in interpreting concentrations and PK parameters in pregnancy. And just one example that we're currently working on in the lab, we had previously published a few years back dolutegravir concentrations in pregnancy, both in the second trimester, third trimester, and then postpartum, and that's shown on the left-hand side. But what we're learning recently is that from that same data set, now we're going back in the lab and measuring unbound concentrations and finding that during pregnancy, unbound concentrations of dolutegravir are higher on average than postpartum. And so looking at total concentrations alone doesn't really tell the whole picture and doesn't give you the best judgment about the reasonableness of individual regimens. So yeah, just a quick plug to consider from an analytical perspective, consider measuring unbound concentrations when possible in pregnancy studies. This is often done with equilibrium dialysis as the gold standard, but can also be done with ultrafiltration. So next slide. So I'll turn it over to my colleague, Ray Suhandynata, who's going to talk a little bit more about the black box, so to speak, and some of the considerations for developing and validating LCMS methods, including in these unique matrices.
Great. Thanks, Jeremiah. Yeah, no, this just, as Jeremiah mentioned, this was originally crafted for a trainee audience. So we've sort of crafted it to be a little higher level, but so just feel free to, if you are an expert in this, I'll try not to bore you too much. So I think a lot of researchers that aren't analytical chemists sometimes think of the LCMS data or mass spectrometry data that's used to generate PK/PD data as a black box. It's just it's a number generator. So today's what I'm trying to do today is to give you a little bit more insight into how the sausage is made, and that's a little joke that Jeremiah and I sometimes have. So let's see. I can get it to switch. Okay, so by far and large, one of the most commonly used chromatography systems is liquid chromatography for bioanalytical analysis. Some people, in the past, it was GCMS, or Greg gas chromatography, mass spectrometry, but most everyone has switched to LC just for the compatibility with the specimens. So just a high level, basically this is a pump system and there's a column that's separating your analytes based off of time, and it's coupled to the detector. By far and large, it's a mass spectrometer. The takeaway here is to let you know that you get something called retention time, and that helps you know that what you're measuring retains on the stationary phase and elutes off that column at a certain time. And if you ask your collaborating labs to export retention time as a data point, you can have more confidence that the data you're looking at is real. So what HPLC stands for is high performance liquid chromatography. That has changed from just liquid chromatography, so we can, it basically works better. And the column stationary phase that I talked about earlier is by a far and large reverse phase. So it's usually a C18 column. So more non-polar analytes bind more strongly. So as I mentioned earlier, mass spectrometry is by far and large the preferred detector. There are other detectors that you could use, but mass spectrometry gives you specificity and sensitivity. And something that I like to remind everyone is that mass spectrometers measure mass to charge of ionized molecules in the gas phase. Generally speaking, small molecules such as drugs are charged only once, but if you are working on biologics like antibodies, for instance, they become multiply charged. So just remember it's mass to charge and not actual mass. Bioanalytical methods for small molecules are performed on generally triple quadrupole mass spectrometers. Those are generally considered the workhorse of this industry. So, and I'll go over a little bit more what that is in the next couple of slides. In this industry, quantitative measurements are gold and typically performed by something called isotope dilution using internal standards. Typically, they're deuterated or C13 labeled matched internal standards. And that's really to address matrix effects, which we'll talk about more, as well as to really understand that the analyte that you're monitoring is real. So for those of you that haven't seen the inside of a mass spectrometer, and specifically triple quadrupole mass spectrometers, is that they're composed of things called quadrupoles, and quadrupoles are simply 4 rods connected together. And I won't get into the details of how this works, but basically this is what it looks like. There's 4 rods set in parallel. They're electrically controlled, and it essentially works as a mass filter. And it pushes ions through the rods, but it also makes sure that the ions that are going through the rods are at a certain mass to charge. And that's kind of where you get your specificity. And this is something that is specific to triple quadrupoles, which is called multiple reaction monitoring. Everyone may have heard multiple reaction monitoring mass spectrometry. And what's happening here is that the ions are getting, the molecules are getting ionized here in the source. And they're represented in this case by 4 different molecules. But let's say you're interested in just monitoring a drug with a protonated M/Z mass to charge ratio of 399. And so what you'll do is you'll set, this is one of those quadrupoles that I showed you in the last slide. And that's what we refer to as the first quadrupole. And it's tuned to let only the ion that's with a mass to charge of 399 through. So as you can see, this blue ion is that represented by that, and it goes through into what's called this the second quadrupole. And it used to be called a quadrupole, and now it's by far has moved on to something called a collision cell. And it's I won't get into the details of that, but this is where you generate something called transitions, and it's basically a daughter ion of the 399 ion. And what we're actually monitoring in multiple reaction monitoring is those daughter ions. So you get extreme specificity and extremely good signal and noise. So the reason why we're able to measure things down into even the low picogram per mil range in samples and biological matrices is because we have direct transmission of the ions through, and we're removing all the noise. And we're moving by the noise by doing a second filtration step here in the third quadrupole by looking only at the ions. In this case, what we would try to monitor is this 223. And then we combine it with that chromatography system that I mentioned earlier to get retention time. So we're looking at retention time, and we're also looking at a specific piece of the whole, if you will, of say this 399 compound. So hopefully that gives you a little insight into what we're doing. So we typically like to start off this presentation by just giving a hypothetical drug, for instance, in milk, maternal plasma, or even capillary heel sticks, and you want to measure this drug. All you really need to know is you need to know the molecular weight. And when you have the molecular weight and you have the structure, then that gives you a great starting point, and then you can start making a method and working with the lab if you're outsourcing that the data collection to another lab. This is typically something that they'll ask for. So The purpose of this presentation is to also give you an idea of what actually goes in. It isn't simply as just turnkey as I've given my collaborator this molecule. Why isn't my assay ready in a month? And it's because, well, we usually can give it to you something in a month, but how will it work? Will it actually be fit for purpose? And I think that's something that I want everyone to think about broadly. For instance, my background as a clinical chemist, we actually validate assays for patient care, and the bar is very high because we don't want to, medical decisions are being made off of that data, and we don't want to be wrong. But for research, it's not that we don't want to be robust and have an assay that's will perform well, but it's a little different bar. And so I just want everyone to think about what's the purpose of the data that you're going to be collecting for. So the things that are important are accuracy, precision, within day and between day. If you're only doing one batch, you probably don't need to do between day, but sometimes it's good to know. As Jeremiah mentioned, limit of quantification, limited detection can be helpful, sometimes not very useful if you're only interested in quantitative data, but if you're doing screening, perhaps limited detection validation is helpful. Sensitivity, which ties into both of those. And as I mentioned earlier, specificity and selectivity, because there's a lot of interferences in biological matrices, and you want to be sure that the compounds that you're looking at are what you think they are. Stability is something else that comes into mind, something that's always at the forefront of my mind when I'm thinking of larger studies, especially clinical studies where the sample collection is being done over months, and then the samples are batch, meaning that you collect some samples that you're analyzing the same day, months apart, and stability matters there. So this is a typical method validation workflow to give everyone just a broad high-level view. Typically, we do something called a pre-validation, which is just a small-scale validation, and it's done rapidly, and the validation kind of is tailored to be fit for purpose, as I mentioned earlier. Research versus testing for clinical care would be an example. And you want to do this because, if you're working with a lab, an experienced lab will always do this, because doing a full validation and finding out there's all sorts of problems, and if you've invested months and months of time, then you just have to start all over again. So we don't like to do that. So that's usually then followed by a full validation, where you get comprehensive validation of all those performance metrics that I mentioned earlier. So to go a little bit more into detail in the pre-validation experiments, typically you'll get something called linearity, and that's usually shown with the calibration curve, meaning that you can perform simple linear regression, and you show that you have a good core coefficient of determination to show that you can, that the response of the instrument is linear. Accuracy matters. So we happen to have certified reference material standards that we know, and we spike those into human samples as well, or have human samples with that have been analyzed by a reference method. In precision, we typically look to make sure that we're not getting wide variability. This is especially important when doing PK studies where you're doing serial sampling. You want to make sure that the longitudinal sampling and small differences between those samples are meaningful. If the variability is 50%, then even those small changes are probably just insignificant and just random error. The LLOQ affects this. So if all your specimens are being analyzed and measured at the lower limit of quantification, then that typically is more imprecise, and that's something to consider about. Matrix effects and recovery. So quantitative versus qualitative. Is this a quantitative assay? If it's a quantitative assay, then matrix effects play bigger roles. If it's a qualitative assay, then you can typically tolerate more matrix effects. And something else is transferability of matrix. Just because it works in plasma, for instance, doesn't mean it will work in milk, dried blood spots, etc. So I find it very common that collaborators will ask us, well, you have this analyte validated in plasma, just do it in bronchiolar lavage. And I tell them, we don't know if that's going to work. And sometimes we have to turn them away unless they're willing to invest the time it would take to actually validate in that matrix. And interferences. Sometimes drugs, for instance, have known interferences that would, and that's because a lot of these triple quadrupoles are what's known as unit mass resolution instruments, meaning that they're accurate to a nominal mass. And sometimes you have isobars in there, and we'll go over those in a little bit. As I mentioned earlier, this is specific really to microsampling. So for dried blood spots, for example, the impact of sample volume really matters. You don't have a lot of sample for, say, heel sticks from a neonate, right? And even this is these are dried blood spot card, and this is typically how, that's the target, right? But even if you go over the target, you're at maybe 100 microliters. And if you don't get a lot of bleed out, then you're at 10 microliters. And the point here is to know that there's variability in the sample collection. At the same time, neonates have a higher hematocrit. You're only going to get maybe 50%. Half of that is actually going to be plasma. So just think about how much you're actually getting out of that neonate. In this case, a dried blood spot's nice because you collect everything, and then you could analyze it by several methods. There's something called paper spray mass spectrometry, which we won't go into, but it's just these are just things for you to think about as you're. There are a lot of challenges with sample collection, especially in this population. You can also extract from these dried blood spots and do LCMS, as we've talked about, as well as some investigators have used actually multi-top MS, which the other thing I'll mention is we actually have all of these, besides the paper spray, we do have the capability, if there is demand for it, to set this up, but we do have both of these available at our core. So going a little bit more high level, again, through the validation parameters. So just not to bore everyone, this will be very quick, and it's also sort of the reference. I know these slides might be distributed, so feel back to look through them and also reach out if you have any questions. So accuracy, generally it's just thought as the agreement between methods, LCMS, MS in this case, and a true value. And our sort of Bible for developing these methods is the FDA Guidance for Industry Bioanalytical Method Validation, which is referenced here. Typically, we analyze a control material of known concentration, and the acceptability criteria for the accuracy is less than 15%, except at the LLOQ, which should be less than 20%. So that gives you an idea of how accurate or a well-validated method should be. And this gives you an idea, just comparing different methods. You have, in this case, on the y-axis with your method, and it's measuring drug X, and this is compared to concentrations measured of the same sample by a comparator method. And the idea is you would want a good correlation coefficient to show that the methods are comparable. But it's also really important to understand the bias. Some methods will have a bias, and that's okay as long as you understand the bias. And that's particularly important if you're using two different labs to compare to do a study for the same sample. The same study, sorry. So if you're using two different labs in the same study and you're getting lab results from both labs and you're comparing the results, you may want to understand the bias. Precision matters, and there's something called within run. So that's within the actual batch, and that's usually less on between run, which is from day to day usually, or even across weeks. And the idea is that this is basically a standard deviation index, and the more variability is in terms of standard deviation from a mean that you have over either specimens in this case. In this case, this would be specimens, but between run as well. It would be important to understand. That kind of brings me next to sensitivity, as we talked about earlier. And that's typically depicted by the lower limit of quantification of a method. And it's also really dependent on something we refer to as signal and noise. And determined by signal and noise, is shown here. So in this case, what do we mean by signal and noise? It's something that I think gets thrown around, but sometimes it's nice to just kind of explain exactly what we mean. So there's a signal in this case for a drug. And this is specifically, it's a transition of that drug, the X that we talked about. And the transition is we're actually monitoring this 138 M/Z ion. And at 10 nanograms per mil of that drug, which is relatively low, but not exceedingly. You have a great signal to noise, 136-1, and what that means is that we integrate the area here, and then we take a look at the noise area, the peak area under the noise over the same amount of integrated area by time. And as you can see, you have a lot more signal than noise. Now, when you're really pushing the LLOQ, you'll actually see that your noise and your signal peaks are kind of similar, and that one increases the in precision. It also reduces the accuracy of the measurement. So it's important if you ever have any questions with a collaborating lab to say, show me your signal and noise at the LLOQ, and you'll get an idea of what you're dealing with. So linearity does matter too. And that's because typically these assays, these quantitative assays, are done using an external calibration curve, and those are typically generated each run. And it's really what it's trying to, when you're asking to assess linearity, it's the ability of your method to quantify an unknown. And so what we typically do is we carry a linearity experiment for every matrix, and that means we would do one for plasma, separate for milk, for dried blood spots, everything because what may be linear in plasma, like an analyte that may be linear in plasma, may not be transferable to the other matrices. Typically what we do for a calibration curve in this case is what I'm showing you here, is that we use a minimum of 5 non-zero points and 5 replicates to show that we can be precise with our calibration curve, as well as a good coefficient of determination. Typically we shoot for something higher than 0.99, the R-squared value, that is. And this is just a note. Typically, you'll see something called 1/x weighting. All that's doing is it's weighting the lower endpoints, which there's more variability there. So typically, we like to make sure we weight the lower calibrators more, especially when we're trying to push the limits of quantitation. So this brings me to the analytical measurement range, and that's, as Jeremiah mentions, the range of concentrations that can be measured directly, and it's always important to know. The reason why is that if a lab is telling you that their range is 0.5 nanogram to 2,000, and yet they're giving you values that are outside of that, either below or above, then you need to question that and ask why, because what the assay is valid is only values around here. So typically when they exceed the range, we typically report it out as less than LLOQ or greater than a ULLOQ, or upper limit of quantitation. So specificity and interferences is next, and this is something that's really important to think about. And it's really the ability of your LCMS method to measure only your analyte of interest. And as I mentioned, there's interferences. So is there anything else that can artificially alter the measurement of the compound that you're trying to monitor? And so typically what we do is, if there is an interference, and we typically look at interferences by analyzing blank matrix, and that we know doesn't have that drug in it. And if there are interfering signals, or peaks as we refer them to, it needs to be less than 20% of the LLOQ of the analyte. So at least it's a fifth. And so it will not impact the LLOQ more than 20% ideally. And interference should cause less than a 15% bias in results. And that's specifically referring sometimes to things like hemolysis, icterus, and lipemia. So bilirubin and lipids in this case. So it depends again on the analytes that you're measuring. Also, isomers or isobars, especially if you're working with mass spectrometry, will interfere, and that's because it's simply massive charge. The mass spectrometer does not see structure, does not see shapes, it just sees mass, and specifically massive charge. So the mass spectrometer wouldn't be able to help you differentiate if you have isomers. And so like L-methamphetamine versus D-methamphetamine, that's actually done by immunoassay in the clinical lab. And isobars, as I mentioned there, these are completely different compounds, yet they have a very similar mass. And if you were using a high resolution instrument, like a high resolution mass spectrometer, similar to what Jeremiah mentioned, then that's something you could do. But with a triple quad, this might be a valid interference. And this is actually an example of what you would actually see. Like, if this is a very extreme example, but for instance, in the left here, this is actually the compound that you're actually trying to measure. you're monitoring this 283.2 m/z ion. And this is another sample where you have other interfering peaks, and it interferes with the shape of the peak. These are 3 different other compounds that are eluting off of the column at similar retention times as the compound of interest. But if your analyst is not really looking at the data very carefully, the software is software. It will just integrate all of this and you'll get a value. So if you see, as an investigator, if you see really anomalous results, ask the lab to look back. So we're all human, we make mistakes, but it's important to understand that the number's only as good as the sample, for one, and also only as good as the method, and also as good as the validation that goes behind that method. So, matrix effects and ion suppression, no two people are the same. Just remember, it's all based off of chemistry. We're just using something called reverse phase chromatography very specifically, and ion suppression in serums, for instance, is very susceptible to phospholipids because phospholipids are they have relatively nonpolar and they tend to elute all throughout what we call the chromatogram. So typically, sort of a minimum, we validate a method by using at least 10 different individuals, and we spike those individuals. For instance, for milk, from 10 different patients, we spike the analyte of interest at the same concentration. We show that we are able to quantitate that concentration accurately in all 10 different patients. If any one patient fails, we typically go back and reevaluate the method. And that just gives us some confidence that it's working and it's transferable to different people. And as I cannot emphasize enough, the method's only valid for the matrices that have been evaluated. So just because it works in plasma doesn't mean it works anywhere else, for instance, or even in milk. Stability does matter. We did talk a little bit about that. It's difficult to do stability because of time and money. Stability studies have to be well thought out. They have to be planned. You have to store the samples. So for instance, if you are collecting samples over 4 years, how do you know your analyte hasn't changed over those 4 years from the first collection to the last collection? Well, the only real way to do that is prospectively know that you're going to be collecting samples, and you would presumably know that if you've designed a study, and to spike in a known amount of your analyte at the starting point of the study and continually monitoring. So at what storage condition you have your actual patient samples at, for instance. And so you test each matrix at every condition that it might encounter: room temperature, 4 degree, -20, -80, whatever. You just have to think about the entire process. And you should also evaluate the stability of all your quality control material calibrators and reagent stabilities as well. Sometimes people have done this for you, so do a literature search, ask your colleagues, ask anybody else that has worked on any of your compounds of interest, because it's likely, if it's a popular one, stability studies have been done. And typically the acceptability criteria there is less than 50% change from the freshly obtained or extracted sample. So I'll just briefly, these are just questions. So as I mentioned, what temperature how long will those temperature samples be stored at? How long can the extracted samples be stored on the instrument? How long can they be run after if you have to re-inject them? How many freeze thaws? And that's all kind of coming into play. So that kind of brings us to a head here. And if you get nothing else, just think about what you can ask your collaborating lab, especially if you're not doing it yourself. Typically, a good laboratory will have this validation summary that's available for your review, especially if you have questions, and they'll have the accuracy, and it usually falls in line with the criteria that I mentioned earlier throughout this presentation. And ask them what their LOQ is. What is it? And then ask them what the range is. And just in case you have data that doesn't really make sense, you can go back to the lab and ask them what the imprecision is, especially if you're doing analysis over many batches over many days. Maybe you have data from a year ago that doesn't really make sense to data that you're getting right now. Ask the lab if they actually are they continuously monitoring their assay. They should, especially if you're trying to use the data from a year ago and trying to directly compare to today data that you're acquiring at the present time. And then matrix effects, it's good to know, and that can help you, especially if you go comparing plasma to even whole blood sometimes has a effect, or even plasma to oral fluid, or plasma to milk, plasma to dried blood spots. It's important to understand how each different matrix changes the result that you'll be obtaining. So that brings us to our summary here. So I hope that you can come away from this talk thinking that knowing a little bit more what we mean by LC-MS/MS. And LC-MS/MS really means tandem mass spectrometry. That's if you see the dash there, there's usually referring to triple quadrupole, but basically you're generating that transition. You're monitoring that fragment ion, so that way you get that extra layer of specificity. Ask questions, work with your analytical lab if it's not your own. Make sure that they validate your assay that you'll be utilizing for its intended purpose, and just realize and have an idea of how much, the more you validate, the more robust the results will be, but the more time it will take, the more money it will take. So just make sure that you're getting what you need, but don't skimp, because bad data, in my opinion, is worse than no data, because it could really send you down the wrong path. And especially in the clinical setting where you could actually be causing patient harm. So bioanalysis in maternal and pediatric clinical pharmacology research poses a lot of challenges. The unique matrices are in itself a challenge, but as I mentioned earlier, and Jeremiah mentioned earlier, is the sample volumes, right? And for instance, if you're doing a neonate plasma study, you don't get a lot of plasma from them that have high chromatocrits, and even if you're not doing a heel stick, the microcontainers typically are only somewhere between maybe 200 microliters to a milliliter, something like that. So that's something to keep in mind. And we've also added all the references in the back, just in case anyone wants to go through. So really the Bible, as I mentioned, for at least in this setting, is the FDA guidance for industry bioanalytical method validation. There's also a lot of CLSI, clinical laboratory standards improvements, documentation that we use in the clinical lab for mass spectrometry methods that are a great reference and these, unfortunately, sometimes you have to pay for, but if you have collaborators that are in a clinical lab, they typically have these on hand. So also the European Medicines Agency guideline on validation of bioanalytical methods is a good one. And also the European Union directive for analytical methods and interpretation of results. So with that, I'll bring us to the conclusion. Thank you for listening. And I'll say thank you on behalf of myself and Jeremiah and all the interests that you have in potential future collaborations and just general questions on education on analytical chemistry and bioanalytical analysis. Thank you.
Great. Thank you both. It's a very interesting presentation with a lot of information. I think that's very applicable to understanding the analytical aspects of pharmacokinetics too. So we do have a question that's been submitted by Murugesh Kandasamy. Says, nice presentation. And when you shift matrix for analyses, do you perform full validation or partial validation when you are within the same linear range?
That's a really good question. So for instance, I think it kind of depends. So for instance, going from whole blood to plasma, we typically just do a partial validation because they're by and large the same. If we're going through, say, from blood to oral fluid, which is just completely different, I would probably at least do the beginnings of a full validation. And if I'm using it for clinical care, then yes, you definitely are doing a full validation. It's a completely different method. So again, this kind of dives into that fit for purpose. Are we just doing research? No medical decisions are being made. If not, then you can have a discussion with that lab and you can do some sort of pre-validation rigorous experiments to show that maybe you don't need to do a full validation. It's still valid within that linear range. So I hope that answers your question.
Yeah, I don't have much to add to that. I think the concept fit for purpose is something we've said a few times and applicable here as well. But thinking about the spectrum of data that are intended to be, for example, included in a regulatory submission to the FDA versus at the other end of the spectrum, maybe preclinical, exploratory, proof of concept type studies. And there's a whole wide gamut in between those, but just thinking about where you fall on that spectrum and what the intention is for the data.
Are there any additional questions? If so, feel free to raise your hand and we can allow you to talk, or ask in the question and answer box. I think I'll have a kind of a similar question on the matrix, and we talked, Jeremiah, you showed the difference between the breast milk and the skim milk, with the THC and the difference there. When you're looking at different matrices from a milk standpoint, do you treat colostrum, fore milk, and hind milk all differently and establish a standard curve in each of those individual matrices?
A good question. We're not currently treating them as completely different matrices, although they do differ quite a bit in composition. But we tend to have a single method, but then consider more on the back end from an analysis perspective as we analyze data and interpret data. Try to consider those things at that stage, but we're not currently developing unique methods for colostrum, for example.
Great. Yeah, there's another question relating to hurdles when you're dealing with milk sampling and how you're collecting that.
Yeah, good question. That's always a challenge, collecting milk samples, and we have in San Diego a milk repository. We work closely with Tina Chambers and others, but often not only the collection of milk samples, but the kind of metadata associated with those samples. So for example, we may know that a lactating woman provided a sample and she was taking a drug, and that's all the information we have. We don't know what dose that person was on. We don't know the last time the dose was taken. We don't know if they were at steady state. So collecting samples is one challenge, and understanding the broader context within individual patients of what their course of therapy was, and then the timing of dose relative to collection, all those things are challenging. And I think interpreting milk data for that reason is complex. But the question brings up a good point, that there's a lot of issues here, a lot of challenges, and we need to do the best we can to inform kind of policy around individual drugs and recommendations of when breastfeeding is acceptable for some drugs or maybe not acceptable for others.
Great, and I will comment here that for people interested in more the clinical aspects and the feasibility of collecting milk samples, they may want to join the workshop we will be holding on January 26th, so next Thursday, on study design in the postpartum period, and part of that will be discussing the lactation sampling in that time frame. So I will put some information and the link to that in the chat for those who are interested. All right, Dr. Choi, I think you should be able to talk now.
Yeah, can you hear me? I have a general question about dried blood spot method. I'm wondering the current status of this method. So for example, how accurately can measure blood concentration using dried spot method?
So just to be clear, it's not something that we've had demand for right now. What I was showing earlier is that we have the capability to implement these methods if the demand were there. So the short answer is we don't know in our hands. The industry's take on the paper spray is that it can be accurate. For instance, we actually have a couple collaborations that are looking at dry blood spot sampling for newborn screening. that is very robust. So I think it depends on the analyte. There's the issues with dried blood spots is getting consistent collection, right? So I hope that answers your question in the sense that we are not currently doing it right now, but if it's something that comes up, we could.
Okay, thank you.
And often we've seen studies that bring forward samples both from venous collection and dried blood spots simultaneously. So from a pop PK modeling point of view, it can be very useful to evaluate sample collection methods as a covariate, and sometimes you can find there are differences, and you can account for those differences from a modeling perspective. But it's something that's necessary to think about early on, rather than just assuming that they're going to be identical to venous blood concentrations or plasma that's obtained from venous blood, I should say.
Yes, it's a very interesting idea. Yeah, I'm very interested in the result. Thank you.
Yeah, I think also with dried blood spots, in particularly the neonate population and even in pregnancy, the variation in hematocrit can really lead to some variation in compared to venous plasma in the concentrations we see. Are there any other questions? I will just ask some clarifying questions about the analytical chemistry core that you run. From a scope of analytes that you work with, obviously you work with a variety of matrices and tissues. Do you only look at small molecules, or are you also able to analyze larger molecules and biologics?
Most of what we've done to date has been small molecules. We do have a long history of performing studies for the Impact Network and also supporting other trials, both industry supported and NIH supported. So occasionally people bring forward projects and serendipitously we say, yes, we already have that assay in place and we're ready to go. But more commonly, it's something that needs developed and validated. Ray can speak a little bit more about our capabilities with large molecules. We do have the instrumentation to do that work, but haven't done a lot of projects so far. So Ray, what would you like to add?
Right. So I think that's probably going to have to be done on more of an inquiry basis, because so for instance, probably the easiest one to do is like a biologic on monoclonal or something like that, because those types of analyses are at least can be standardized, and typically they will be done by digesting the monoclonal antibody and matching monitoring peptides that are specific to it. We've also had some inquiries that are more on the research side, looking at peptide drugs, things like that. Those ones are highly dependent on how well that peptide ionizes and chromatographs. So I don't want to overpromise and say that, yeah, we can do that, but I don't want to discourage anyone from reaching out and starting a conversation, because I think it, as Jeremiah mentioned, we do have the capability and expertise to do quantitative measurements there, but sometimes it's easier said than done.
I agree. There's definitely some complexities to measuring the biologics that we don't do in our small molecule cores either. So,
yeah.
Right. All right. Well, we are nearing the top of the hour. I do have one additional question to say, sorry, I joined the webinar late. What was the 5% matrix enhancement that you were discussing?
Oh, that was just an example. It's good to know how much matrix enhancement or suppression you have. So that's, I think we had to remove some of the slides that actually show the calculation to show how much matrix enhancement you have. Typically, you'll just do it with a blank to show that, well, in solvent, this is what I would actually see, but in say plasma, for instance, we get a 5% bias up, and that we can attribute that to the matrix. I hope that clarifies that.
Great. If there are no additional questions, thank you very much for the talks today. I know it was very interesting and informative, and hopefully will clarify some aspects of analytic drug analysis. Thank you.
Thank you.
Thank you.